Good afternoon. It's a pleasure to be here. So, Sigilon's a new company. It's been in existence for just under a year, but I want to highlight sort of who we are and then talk a little about the technology and where we're going. Get all the builds here. There we go. So, Sigilon, who we are, was founded out of technology, coming out of the Massachusetts Institute of Technology. You can see the board of directors there. It's a good mix of folks from both the venture world, but also from former industry folks as well. So it's a good mix of folks. Very strong board of directors. See the management. Paul Watton, who's our CEO, was CEO of Okada, which was purchased by Astellas about just over a year and a half ago, or a year ago. So, world-class team at Sigilon. We're very excited to have the team we have, and even more excited about the technology. So we are funded by Flagship Pioneering via their approach they take to incubating and transforming companies. So very excited to have them as our investor, sole investor at this point. So what is Sigilon? So we view it as game-changing technology that hopefully can move us to sort of the next generation of cell therapeutics. So the concept is really to be able to encapsulate cells and then implant those into the body to produce whatever you'd like. So therapeutic enzymes, factors, antibodies, hormones, you name it. So it's bringing the manufacturing basically out of CHO cells, put it into human cells, and actually putting it in the body. So the value proposition obviously is, you can imagine the benefits here. Your dosing is going to be redosed much less frequently. Broadly applicable to a wide variety of molecules, and you can deliver proteins for either diseases that are treated today but not optimally, or those that have not been able to be treated before due to low, really short half-life proteins or other problems you may have. So what's the platform of the technology? So the platform is really around two pieces that then combine into really the product. So you've got a proprietary cell line approach. So using human cell lines, engineering those cell lines to produce your factor or protein of interest, combining that with a novel encapsulation chemistry to then have your final product, which you can then put into the body. So the cells are protected from the immune system by the encapsulation technology, which I'll talk a little bit more about. We do believe it's broadly applicable. Should be relatively simple to administer, and we believe that you should see significant durability and the ability to redose. See if I can move it to. So as many of you know, one of the big challenges in trying to implant cells into the body to do anything has been the immune response. The foreign body response rapidly kicks in, either destroys the cells, or if it's a device of some sort, builds a fibrotic capsule around it and subsequently renders it unusable. See some pictures of that here. So what this technology is built on, there were a series of Nature publications that came out that really built out this, what we're calling super biocompatible materials, so this novel chemistry. And I'll talk a little bit about some of the data just to give you a flavor for it. So first, there's a whole series of proprietary chemistry that underline this that allow you to control host immune responses. So effectively rendering the whatever you put in, the material you put into the body, almost blind to the immune system. So you're not getting these fibrotic responses, which is critical to our success. And I'll give you a couple examples of the data. So this first example, so this is looking at two different materials that have been encoded. So PDMS, which is used a lot in many different medical devices, or medical grade silicone. And you can see on the top on the left-hand side, so these have been put into an animal for 2 weeks. You can see the fibrotic response on the top, which is quite significant in just 2 weeks. And these were not modified. These are just PDMS or medical grade silicone stuck into the animal. Down the bottom, you can see the, and these were discs that were put in. You can see the discs that have been coated with the Sigilon technology, and they come out clean. So what about if you begin to look at what's going on from a cellular perspective? You look on the right-hand side, you can see on the control side again, significant cellular buildup around the materials. And you can look, this is various staining approaches we've taken. We've looked at a whole host of various immune cells and different molecules to really get a readout on what's going on and understanding the mechanism here. But effectively, the net is that the immune system doesn't see it. It's effectively blind to this the novel chemistries, and thus you don't get the massive fibrotic response you see. So then the question is, well, how does it look in the body? So in this case, we moved to a primate. So we took a couple of different approaches that have been taken previously for encapsulation materials and compared them side by side against our materials. So these were done in primates. The Sigilon capsules are on the left-hand side, and you can see them. And this is after 4 months in the primate peritoneum, just to give you perspective. You can see the Sigilon capsules look like they were just implanted, just put into the peritoneum. The right-hand side, you can see the massive fibrotic response that occurred with one of the comparators we used. And if we look at, we got one more. In fact, when you after 4 months you pull them out, you can actually just there's no clumping. You can actually, with gentle washing, just remove the capsules right off the omentum. So quite amazing in that at 4 months you see effectively no fibrotic response or foreign body response against the capsules. And these are empty capsules. Now what happens if you put cells in? So this first experiment, so this is looking at glycemic control in immunocompetent mice. So this was done in the C57 black mouse strain, which is probably one of the most rigorous fibrotic models out there. These mice exhibit a very strong response to any foreign body. And in this case, these are xenogeneic islets. And they're encapsulated in the Sigilon technology. And then this is a graph of the blood glucose measurements over a 6-month time period. So you can see the healthy mouse in green, and then you can see the Sigilon implanted islets in the kind of a brownish color. And you can see that effectively over a 6-month period, you're basically able to nearly mimic the blood glucose effects you see in the wild-type mouse. Now, why that's significant, typically when you implant any sort of islet, whether it's encapsulated or not encapsulated, you'll see a response over a very short period of time, then it goes away because the cells are either destroyed or you get a fibrotic response that encapsulates whatever material you've put in. So this was very encouraging to us. So then we took one other approach and said, okay, so we've seen this with islets. What about if we take another cell type? So in this case, we just took 293 T cells and actually engineered them to express EPO. So EPO-eaten. And then we put them into a hard device. You can see the encapsulation on the top into a hard device. And then implanted those into the mice. And then looked at your serum EPO levels as well as your hematocrit as a percent of the body. And this is was a really interesting experiment because if you look down on the lower left, so this is the serum EPO levels in the mice. And you can see that effectively using 3 arms, so a mock device, so an empty device, an uncoated device that contains the cells, or the Sigilon device, coated device, which contains the same cells. And you can see that on the uncoated device, you get an initial response for maybe the first 5 to 10 days, and then it slowly peters out as the fibrotic response hits. On the Sigilon device, you can see the cells continue to build production and produce high quantities of EPO. If we flip to the right-hand side, you can see those same arms. In this case, you're looking at hematocrit. So the mock device is basically going to mimic what you would see in a normal mouse hematocrit. The uncoated device, you can see, diverges at first, but immediately comes back in line with where the mouse is. And then the Sigilon coated device, you can see, continues to produce significantly more EPO over time. Now, obviously, EPO is a good proof of principle for the technology. You can begin to imagine that getting people's hematocrit up to 70% is not a therapeutically advisable strategy. But for us, it was very, it's good proof of principle because you can begin to look at all sorts of secreted factors, proteins, enzymes in the body that you can produce that have a relatively wide therapeutic index. If you take the case of hemophilia, for example, where if you can bring a patient's levels from a severe patient that has, producing between 0 and 1% of normal factor 8 or factor 9, you can bring that level up at a sustained level to 10, 20, 30% or more of normal. It's effectively curative for those patients for the most part. So where are we going from here? So we feel like we've got excellent proof of principle of the technology, both in primates and as well as in rodents. We've got additional experiments that have been run beyond what you see here, using different cell types, different animals, different factors, and are very excited about where we're going. And from here, it's really about establishing the right cell line that we want to use and moving forward in our lead indications to the clinic as fast as possible. One other thing we like about this technology is that because you're dealing with, if you deal with a cell line that's been in humans before, then you're dealing with a factor which is obviously treated today. Your risk, you lose that target risk. You've also downplayed a lot of the risk on the cell line itself from a regulatory perspective. So we like it in that it's a lower risk approach to entering this space where you can have significant therapeutic effects and impact on patients' lives. So with that, thank you.